we started with cell division, and we saw a little bit on this cell division. To summarize, vamos allá. We went through all these structure, number of chromosomes and chromosomal abnormalities, and we saw cell division for prokaryotic cells, which was the easiest one. We said that in prokaryotic cells, when they want to, that's the transmission of genetic material. When cells, prokaryotic cells, want to multiply, that is their aim, their objective would be produce more organisms because they are unicellular. The only thing they do is they just duplicate the genetic material that, as we said, is always attached in one region to the cell membrane. And then after multiplying this genetic material, half of it will go to one new cell, the other half to the other one, and they undergo this binary fission. So if I ask you to explain the cell division in prokaryotic cells, you would have to mention just 3 things. First of all, the objective, that is always generate a new organism. So coming from one bacterium, at the end we've got 2 bacteria. Second, that we've got, as it always happens, first of all, the duplication of the DNA. And then, this DNA is always attached to the cell membrane, and at the end we've got this binary fission that is breaking into binary because we break in 2 pieces. That's the process by which prokaryotic cells divide. So we've got this presentation here that explains more or less what happens here. We've got this original DNA that would be the light blue. Then this one duplicates and forms another molecule of dark blue DNA. Then we've got this division, and at the end we've got 2 identical bacteria. So that's how cell division in prokaryotic cells happens, and is not difficult at all. But we had to start with this process of cell division in eukaryotic cells right now. If you go back to this section in which we said how and why do cells divide, that is at the beginning of the unit, this one, how and why do cells divide, we said that this answer to the question why depends on whether I'm talking about a unicellular or a multicellular organism. In the case of bacteria, there's only one answer because every bacteria is unicellular. But in the case of eukaryotic, the answer for the question why depends on whether this eukaryotic organism is unicellular, or in the case of humans, we are multicellular. So does cell division happen in my body? Yes, it's happening every second. What is the reason for it to happen? Why do I do cell division? Either to grow or to repair the structures. Whenever this is needed in my body, whenever I need to grow or I need to repair a structure, a process of cell division is going to happen in my body. Does it mean that cells are always dividing? Well, again, is one of these tricky questions. There's always cells dividing in your body. There's always some cells that are dividing in your body, but not every cell, obviously, is dividing in your body. Remember that we're going to talk about 2 structures formed by our DNA. When the cell is doing its normal job, what is the structure that the DNA is in which form is the DNA when the cell is working, doing its normal job? In the form of DNA in the nucleus when the cell is working is in the form of, anyone?
Chromosome.
Chromatin.
Chromatin.
Okay. When the cell is working, your cells are going to have in the nucleus this structure that is chromatin. Almost always in your cells, you're going to have this DNA in the form of chromatin because most of the time your cells are going to do their job. What is a cell in your heart doing most of its life? Contracting in order to pump blood. So most of their life, they are going to have their DNA in the form of chromatin because they are going to be working. But there's going to be a moment for this cell in your heart in which is going to receive a message, is going to be receive this order. You've got to duplicate. During this short period of life, your cell will transform this structure, chromatin, into chromosome and will divide. Okay, this division is known as mitosis. Mitosis is a term that we can use for two different things: division of the cell, but we usually refer to especially the division of the nucleus. Okay, so we talk about mitosis for the division of the nucleus and then cytokinesis for the division of the cytoplasm. Remember that when we mention this part, how cells divide, we said that first of all they multiply the genetic material, then they divided the nucleus, and at the end they divided the cytoplasm. So this division of the nucleus, the second stage, is mitosis. But we're going to see now all the sí, que está representada. We're going to see now all the stages in the life cycle of a cell. The life cycle, one cell, when we look at it in the microscope, one cell could either be found during this process in which is carrying out all its job, all its metabolic processes, then my cell is going to be considered to be in interphase. What is the definition of interphase then? Is this period of time, this phase, this moment in the life cycle of a cell in which the cell is not dividing. What is the other phase then? Cell division. My cell could either be dividing, is in the phase of cell division, or not dividing, is in the interphase. It would be very easy if my cells could only be in these two phases, but before the division, I need the cell to be prepared for the division. So during the interphase is not only that the cell is going to be working, there's also going to be another part of this interphase in which my cell will be preparing for the division. So my cell could be either working, preparing for the division, that is interphase, or second option, being actually dividing, that is the mitosis. Okay, we're going to see this in a diagram. This diagram here is representing the life cycle of a cell. So one cell that has already been born, that has already appeared, is going to be here in this section. This is when cell division happens. This cell has just appeared. We are here. What is the first thing that my cell that has just been formed, what is the first thing that this cell is going to do? We said here, no, okay. We said when we talk about how cells divide, a ver si voy a esa y lo veis bien. When we said these last sections, how cells divide, we said that the duplication of the DNA was always equal, always the same, exactly the same genetic material in both new cells. But the division of the cytoplasm could not be identical. So when the cell has just appeared, what is the first thing that my cell is going to do? Complete itself. So complete the number of organelles that it needs, the number of mitochondria, the size of the endoplasmic reticulum, complete all the cytoplasm, and if needed, increase a little bit the size. That is the first thing that my cell is going to do. Now that it has the right size and the right components, what is it going to do? Working. If it's a heart cell, it's going to start contracting. If it's a liver cell, it's going to start producing bile. If it's whatever, depending on the type of cell, it's going to start doing its job. That is the second thing. Is it division? Not yet. So because it's not division, that is going to be part of this interface. If you look at this picture, this diagram, is representing the same as the previous one. Previous one, interface, cell division. Next one, in a circle, interface, all that in blue, cell division or mitosis for the nucleus and cytokinesis for the cytoplasm. This is all the cell division. So what is it representing? That my cell could either be in the interface or dividing. Which process seems to be longer to last more time, interface or cell division?
Interface.
Interface seems to be longer. My cell most of the time is going to be either growing until it has the right size and the right number of organelles, then performing all the metabolic processes. That is going to take a long time, and then my cell will receive this message. You need to duplicate. Then the next thing that is going to happen is that my cell will prepare for this division. How is it going to be prepared? Well, first of all, replicate the DNA. So each of these 46 chromosomes that human cells have, instead of having one chromatid, in this phase, S phase, they will turn into something with two sister chromatids that are identical. That happens here in the S phase, but the cell is not dividing yet, so I'm still in the interface. How do I differentiate this part of the interface from the other one? Well, just giving a different name. First part of the interface is going to be G1. We call it G because apparently nothing was happening to the cell. So it's gap. Gap is an empty space, so it was considered that nothing was happening. The cell was doing its normal job, okay? Then the second phase in which we synthesize, we form new DNA is the S phase. We are going to see that it's visible that the amount of DNA is going to turn into something, into a double amount. Remember, still 46 molecules, but those molecules will contain double size, double information, because each chromosome will have 2 chromatids. Then we've already have the DNA, but we need all the components that are going to allow this division. I will need especially enzymes. Remember this term, enzymes, that were chemical substances. We know them from digestion because they helped us digesting food. ¿Os acordáis de chemical enzymes that we saw last year? Well, we're going to have enzymes here as well that are going to help me in the division, increasing the speed of this division. When do I form all those enzymes? Just before starting the division. So I'm still preparing for this division. S phase, you are preparing duplicating the DNA. G2, you are preparing forming every structure that you need for this duplication. Okay, so that is interphase. Interphase consists on G1, the cell is either growing or doing its normal role. S phase, the cell is duplicating the DNA, and G2 is getting prepared for this division. G2 finish when mitosis begins, when it starts. Well, because scientists love giving different names to different processes, you already know it, interphase, mitosis, cytokinesis, G1, S, G2, they also gave different names to every stage in mitosis. Okay, we're going to see those names. They are difficult terms because it's loads of new names, but do not panic. We're going to see it today, we'll see it tomorrow later, and you will see that it's not as difficult as it seems at the beginning. So cell division, as we mentioned, consists on mitosis, that is division of the nucleus, and cytokinesis, which is the division of the cytoplasm. These are all the strange names in mitosis. We've got all these names here, but do not panic. I want to show you, first of all, this video, but I need you to tell me whether you still hear me while I'm launching the video, because I'm not sure whether you do. Vale. ¿Me oís ahora mismo aunque esté el vídeo puesto? ¿Me podéis?
Sí, sí.
Cuando estaba puesto también, ¿no? No solo cuando lo he parado, sino cuando estaba puesto también. Vale. Okay, what are we going to see in this video? Because I don't need the sound, it's just music, what you've got in the video, so it's not important. What am I going to see here? We're going to see the camera getting inside a cell. So what we've got here, that is one cell, that is another one, that is another one. And if you pay attention, what is going on there is that some of these cells are going to divide. Okay, we're getting inside the cell. So what do you think is this structure here inside the cell? What could it be? Any idea on one structure inside the cell that has this shape? A ver si lo veis mejor en esta, que se ve regularcillo.
A mitochondria?
It's a mitochondria, okay? One mitochondrion, that would be another one. And we are going to see different organelles in the cell as well. That's a mitochondrion there. What do you think is here? We've got one structure that is sending vesicles. Any idea?
The Golgi body?
That's the Golgi body. And this Golgi body is connected to the rough endoplasmic reticulum that we've got here, and that is a nucleus. Okay, we'll see it. As you can see, this Golgi body is receiving vesicles from the endoplasmic reticulum, sending them out. They are connected, and that is the nucleus of the cell. And what we're going to do right now is we're going to get into the nucleus through this. What is that? Nuclear pores. Okay, so nothing ever gets into the nucleus apart from us. We are going to get into the nucleus, and what do you expect to find in the nucleus? ¿Qué vamos a encontrar when we get into the nucleus?
Chromatin.
The chromatin. And you're going to see how this chromatin turns into chromosomes because our cell wants to divide. We're getting inside the nucleus, and you see a central structure there that must be the nucleolus, and we've got chromatin there that is going to condense in order to form the chromosomes. As you can see, we are in the interface. Interface means that the cell is either working or getting prepared for this division. So the DNA is going to be in the form of chromatin, no chromosomes, because chromosomes only appear when the cell is going to divide during this cell division. You've got there the nucleolus disappearing and chromosomes being formed. Well, what you've seen right now is that the nuclear membrane has disappeared. Bueno, aquí llega mi primera confesión. I've told you loads of times that the nuclear membrane is always protecting DNA, but it's not true. When the cell is going to divide, for a very short period of time, the nuclear membrane disappears. Because it disappears, this structure here, this one in the cytoplasm, could be connected with the genetic material. What do you think is this structure here in the cytoplasm with this T shape?
The centrioles.
The centrioles forming the centrosome. Okay, so you're going to see how this centrosome connects with chromosomes in the nucleus that are already free because the nuclear membrane has disappeared. Okay. We've got one centrosome there, another one in the opposite pole, and you've got these aster fibers that will be joined to my chromosomes. Okay, so we've got two centrosomes, one on one side, the other one in the other side, and they are forming this structure. What was the name of this structure?
Mitotic spindle.
Mitotic spindle. Okay, so thanks to this centrosome that has duplicated, each one went to one pole, we've got this mitotic spindle. Bueno, there's one mistake in this video. I like it a lot, but there's one mistake, because it's not that the chromosomes magically go to the center of the cell and then the fibers join to them. In fact, the fibers will join two chromosomes first, and it's the fibers that put them in the central parts, ¿vale? Pero bueno, por lo demás, more or less is okay. Well, we've already seen two names in that lower part of the screen. You probably didn't realize. The first one was prophase. The second one appearing is metaphase. I'm going to go a little bit back for you to see this. When the nuclear membrane disappears, one phase known as prophase starts. Then we've got, in this video, you've got pro-metaphase and metaphase. I'm not going to use 5 names, but just 4. So from prophase, we will go to metaphase, pero iré más despacio en eso, no os preocupéis. So you've got pro-metaphase, then when all of them are in the middle, in the moment in which all the chromosomes are in the middle, as long as the chromosomes are in the middle, we are in the metaphase. So it's easy to identify. If you look at a cell and you see all the chromosomes in the equator of the cell, se llama así el centro, el ecuador, in the equator of the cell, then you know that you are in the second phase of mitosis. First phase, what is it going on in the first phase? The nuclear membrane disappears, the chromosomes become visible, the centrosomes go to different poles of the cells. What happens in the metaphase? Just that the chromosomes are in the middle. So now we've got all the chromosomes in the middle, and these fibers joined to this part of the chromosome. Name of this part?
Centromere.
Centromere. So these fibers will join to the centromere, and what's the following thing that is going to happen? That the fibers will get shorter, so they will start pulling and separating the two identical parts of the chromosome. Let's see it. They pull and they separate these chromosomes in two. As they start separating, we've got a new name here. That is anaphase. That is the third phase. First one, prophase. Second one, metaphase. And then when they start separating, we've got the anaphase, the third phase of mitosis. Now, we already have two sets of genetic material, two different sets of genetic materials in each pole, in each part of the cell. So what do we need? We need the nuclear membrane to reappear. That's what has happened here. One nuclear membrane and another nuclear membrane. For a very short period of time, my cell is going to have two nuclei. But what do I need after that? The division of the cytoplasm, so the cell breaks in two. Going back to this picture, here we've got all the division, and once I have my nuclear membrane there and the other one here, my cell seems to have two nuclei. But I'm going to separate that into cells. That's what's going on in this stage of the video. A ver ahora cómo vuelvo yo al vídeo. ¿Dónde estaba eso? ¿Dónde veo yo eso? En esta, perdón, que tengo tanto abierto que ya no sé. So, as you can see, the components in the cytoplasm could either go to one new cell or the other one, but there's no problem. They will reappear later at the beginning of the interphase, the calm phase. What's the name of the last phase in mitosis? Telophase, because we've already said that telo means the end. At the end of the play, bajamos el telón. The end of the chromosomes were the telomeres. The last phase is the telophase. Okay, so going on, after this telophase, what I'm doing here is I went out of the cell, and now the cell membrane will split in two. The cytoplasm breaks in two. What we are seeing here is more divisions, because every moment in my life, my cells will be dividing, okay? So that's what we're going to see little by little, this process of cell division. But it's very important that you understand this is continuously happening, and it's only humans, it's only scientists that in order to study that, we call the first stage prophase, second one metaphase, third one anaphase, last one prophase. But the process is continuous, as you saw it in the video. Okay, it's very important that you understand that it's a continuous process. It's only that we give different names for different stages. Okay, bueno, then mitosis, division of the nucleus, results in the formation of two daughter cells, daughter nuclei. This mitosis is divided in 4 phases, and then cytokinesis, that is the division of the cytoplasm, that is going to start when the mitosis is just about to finish, and it will result in the formation of these 2 daughter cells. Well, these are all the stages that we saw in the video. In the video, you've got prophase, prometaphase, metaphase, anaphase, and telophase. But I told you that this year we are going to see these 2 as a single phase. So we'll name only 4 phases. First one is prophase, second one is metaphase, third one is anaphase, last one is telophase. Scientists sometimes use 5 stages because there's a lot of things happening in this prophase. So as we've got loads of things, they prefer to give 2 stages for this one. But in the Spanish book, you've got just 4. Next year, you're going to see just 4. So I prefer to give you 4: prophase, metaphase, anaphase, and telophase. Okay. What we're going to see later is what happens in each of these phases. What happens in the prophase? Pues vamos allá. Prophase, as we said, is the first part of cell division. Okay, we've already seen it. What happens there? That the centrioles forming the centrosome, the centrioles will go to the opposite poles in the cell. So in G2, centrioles from one, centrosome from one, they form 2, but they remained in the same position. It is during prophase that they migrate because they come from an original structure, and this one duplicated, and we had the aster fibers in the middle. You've got to think as chewing gum, so sticky proteins. If you separate this chewing gum, in the middle you're going to have a sticky substance, which is going to be my mitotic spindle in the middle. Then these centrioles migrate to different poles, and therefore they form this mitotic spindle. This another thing that happens is that the DNA is going to appear as chromosomes. So if you see in the microscope a cell and you do identify a thick DNA, so the beginning of the formation of these chromosomes, you could say that your cell was undergoing prophase, the first one. Chromosomes start being visible. And the other thing that happens is that the nuclear membrane disappears. It breaks down and disappears. We saw more things like the nucleolus disappearing. We saw it in the video. But you've got to learn these 4 things that happen in prophase. Is the first part of the cell division. Centrosomes go to opposite poles, forming the mitotic spindle. DNA appears to be visible, forming these chromosomes, and the nuclear membrane disappears. We've got it in this picture. This is the nucleus, and this is the nucleolus that is still there. Do you see any chromosome there? Can you see any visible chromosome?
No.
No, you can't. So which phase in the cell cycle is this cell in? It must be before the division. So that is interphase. That is still interphase because you don't see the chromosomes that are visible. Then why do you have these two centrosomes there? Because it's the G2 in the interphase. So you are already prepared for the division. What is going to happen next? That these centrosomes will migrate to the poles of the cell. As they migrate, the mitotic spindle starts being formed. Can this mitotic spindle connect with my DNA? No, because there is a nuclear membrane there. So what is the next thing that I need to do? Make this nuclear membrane disappear. Other things that are happening in this phase is that the DNA is already visible. If you see the chromosomes, you are already in prophase. But we've got here early prophase and late prophase. In the other diagram, you had prophase, prometaphase. Prometaphase would be the late prophase. But this year I told you that prophase is enough. So what's happening in prophase? This mitotic spindle is formed, the chromosomes are visible, and what's going on? What's the difference between this picture and that one? That the nuclear membrane has disappeared. We've got some fragments of the nuclear membrane there. Because there's no nuclear membrane, my fibers in the mitotic spindle can connect to different chromosomes. As time goes by, these chromosomes condense more and more, so they are going to be more visible and they are going to be a little bit shorter. That's another difference between this picture and that one. Okay, these fibers connect to the chromosomes. What are they going to do with them? Put them in the equator, in the middle. As long as I have them in the equator, in the center of the cell, in the metaphase plate, el plato de la metafase, in the equator, I already have the metaphase. So as you can see, this phase is very simple. What happens during the metaphase? That the chromosomes are aligned in the center. Okay, that's like the goal. Es como la meta. Acordaos de eso también. You've got this goal, la meta, visible. That's metaphase. This line in the middle. It's in the middle of the division. So as long as you see these chromosomes aligned in the middle, you can identify this as metaphase. As you see, each chromosome has these two sister chromatids with identical genetic information. During the metaphase is when we usually take the picture of the cells in order to build the karyogram, okay? Because they are shorter and it's easy to identify all the fragments, all the bands in the DNA. What's the following stage, the third one? Vuelvo un momentito para atrás. We said prophase, metaphase. The third one is anaphase. What happened during anaphase? That the chromosomes separated and they formed this shape. This shape that is an A, isn't it? It's an A moving to the opposite pole. So that is anaphase. That's what we've got in this following phase, that the chromosomes are pulled apart so they will go towards each opposite pole of the cell. The cell starts getting a little bit longer in this phase. You've got it. That is not round shape, but it's getting a little bit longer. This one is the anaphase. In anaphase, my chromatids are already independent. So we start calling them, instead of chromatids, we start calling them daughter chromosomes, because they will form part of this new genetic material in the daughter cells. So they are daughter chromosomes. What is the last phase? Final in Greek was, como decíamos, final in Greek?
Telo.
Telo. So what's the name of the last phase? Is telophase. What happens during telophase? That my chromosomes that were visible, they are going to start uncoiling, so they are not going to be condensed, but they will decondense, se desespiralizan, uncoil es eso. They become chromatin again. The nuclear envelope will appear again, and the spindle will disappear. So it's like the opposite to the first stage. In telophase, this nuclear membrane will appear again, my chromosomes will not be visible, my nucleolus appears again, and the mitotic spindle disappears. After this phase, or more exactly, during the end of this telophase, cytokinesis starts, which is the division of the cytoplasm. Okay, so those are all the stages in mitosis. The last one would be the cytokinesis, that is these cells breaking down into two daughter cells, two new cells. Okay, well, it's 12:15, and I've prepared for you in your virtual class one of these interactive videos that you can either do right now or maybe just relax and do it this afternoon as you prepared. You've got it in this unit, Unit 5, Cells, the building block of life, in this section, actividades, ¿vale? In contents, en contenidos is what you have, the PowerPoint presentation and all the videos. But if you go to the section actividades, you've got this interactive video that is just a review on today's class. And it has, I think it's 6 different questions, okay? It's not a very long video. Let's see how long it is, how much it lasts. A ver, dice aquí que is 6 minutes video, so your homework will take more or less 10 minutes, ¿vale? Te lleva, because it's just answering the questions that they're going to appear